Okay, so welcome back to this next video in which we are discussing phospholipase C gamma enzymes. Okay, right, so we've now discussed the reaction which phospholipase C enzymes catalyze, which is this breakdown of phosphatidylinositol 4,5-bisphosphate into diacylglycerol and IP3. Okay, right. So we now want to discuss the different families of phospholipase C enzymes, and then we'll focus in on the phospholipase C gamma family of enzymes. Okay, so firstly, the different families. There are 6 different families of phospholipase C enzymes. Okay, the first is the phospholipase C beta family, which contains 4 members. Okay, it contains phospholipase C beta 1, then you've got phospholipase C beta 2, phospholipase C beta 3, and phospholipase C beta 4. Now, when people generally think about phospholipase C, generally they will be thinking about a phospholipase C beta family enzyme, okay? Because these 4 enzymes here are the forms which are going to be activated by GQ/11 heterotrimeric G proteins. Okay, so GQ G proteins interact with these phospholipase C enzymes. Okay, so they're the main forms of phospholipase C that people generally know about, because generally they know about phospholipase C being downstream of G protein coupled receptors such as the alpha-1 adrenergic receptor, the M1, M3, M5 muscarinic acetylcholine receptors, etc. Okay, so that's the main family that people generally know about. And the next family is the phospholipase C gamma enzymes, which are the topics for this video. Okay, and this contains phospholipase C gamma 1 and also phospholipase C gamma 2. Okay, so these are the 2 enzymes within this family. Okay, and these are the stars of the show. These, as we'll see, are going to be activated by receptor tyrosine kinases. Okay, so they're not activated by G protein coupled receptors at all. They're activated by a totally different family of receptors. Okay, but they catalyze the same reaction, the breakdown of PIP2 into diacylglycerol and IP3, and they cause the release of calcium just as a GQ coupled, G protein coupled receptor would. So it's the same pathway for a totally different family of receptors. Okay, right. Next up we have the phospholipase C delta family of phospholipase C enzymes. And in this there are 3 members. Okay, so there's phospholipase C delta 1, there's phospholipase C delta 3, and there's phospholipase C delta 4. Okay, then the next family is the phospholipase C epsilon family. There's only 1 member within the phospholipase C epsilon family, which is phospholipase C epsilon 1. After that, there's the phospholipase C eta family. Okay, so we're going to more and more elaborate Greek symbols. Okay, next, so there's phospholipase C eta 1 and there's phospholipase C eta 2. So there's 2 members of this phospholipase C eta family. And then finally, the final family is phospholipase C zeta. Okay, and there's only 1 member of the phospholipase C zeta family, which is phospholipase C zeta 1. Okay, so just in case your Greek alphabet is lacking, okay, this is the Greek letter eta, and this is the Greek letter zeta. Okay, right, so that you can go and look them up and practice drawing them. Right, so we're going to focus on the phospholipase C gamma family, which contains these 2 members here. But overall, there are 13 phospholipase C enzymes shown here. 4 in this family, 2 in this family takes us up to 6, 3 in this family takes us up to 9, 1 here, 10, 11, 12, and then 13. Now, these are all separate genes. Okay, each of these genes in some cases has multiple splice variants, okay, so that adds to the complexity. Right, so we're now going to focus in on phospholipase C gamma enzymes. Okay, we want to look at their structures and we want to look at how these two forms of phospholipase C gamma are both activated by receptor tyrosine kinases. Okay, so they both of them have pretty much the same structure. Okay, and they're also both activated by receptor tyrosine kinases. So what we will discuss is general for both of these forms of phospholipase C gamma. Okay, right. Now, you will struggle to find this information within a textbook. Okay, so I'm going to reference the paper I got it out of. Okay, and the paper I got this information from is Gressett et al., and that was written in 2010. Okay, so the primary author was Gressett, but there were a bunch of co-authors as well, and it was a paper written about phospholipase C gamma enzymes and their mechanism of activation in 2010, so that's well worth a look. Okay, and that's the source of my information. Right, so let's now move on then to the structure of phospholipase C gamma enzymes. So the first thing to say is that we do not actually have the crystal structure of phospholipase C gamma enzymes yet. Okay, however, we do know their primary structure, and we also know the crystal structures of phospholipase C delta enzymes and phospholipase C beta enzymes. And the primary structure of phospholipase C gamma enzymes is almost identical to the phospholipase C beta and delta enzymes. So we can hazard a very educated guess at what the three-dimensional structure of phospholipase C gamma is. And that's what I'm going to tell you about in this video. Okay, so we'll start off with the primary structure, and this isn't guesswork. Okay, so we have the amino terminus of the protein here. So each phospholipase C gamma enzyme is just a single polypeptide. They're not complexes of polypeptides, it's a single polypeptide. So here's the amino terminus of that polypeptide. Now the first important domain that you have within a phospholipase C gamma polypeptide is a domain known as a plextrin homology domain. Okay, and for short, plextrin homology domains are often abbreviated to pH domains. So we can abbreviate this to pH for plextrin homology. Okay, right, so this is a domain that you find in a huge number of different proteins. Okay, so it's just a conserved structure that you can find all over the place in many different proteins. And it was originally found in the protein plextrin, which is important in platelets. Okay, so that's where all of these domains since have been named after that domain that's present in plextrin because they are homologous to it. Okay, which just means similar. Okay, so this is just a domain that's similar to another domain that you have within plextrin, and you find domains like this in many different proteins. Okay, so that's the first interesting domain that you have within the primary structure of this phospholipase C gamma enzyme. Okay, now it's not going to be hugely important in the activation of these phospholipase C gamma enzymes by receptor tyrosine kinases. However, there is more information on this subject than I am going to present to you. There are other ways that phospholipase C gamma enzymes can be activated, and in those other ways, plextrin homology domains are important. Indeed, this plextrin homology domain is important. Okay, but it's not going to be important in the way that receptor tyrosine kinases directly activate these enzymes. Okay, so we're not going to talk about that hugely. Okay, following the plextrin homology domain, you have three domains which are known as EF hand domains. So I'll draw, four of these EF hand domains. Okay, now EF hand domains are usually domains that are extremely important in calcium sensing proteins. So the sort of archetypal protein that has four EF hand domains, just like all of these phospholipase C gamma enzymes have, is calmodulin, which is a very important calcium sensor within the cell. Okay, now in calmodulin, these EF-Han domains, they all bind calcium, so a calcium binds to every single one of them. Okay, but in phospholipase C enzymes, the EF-Hans do not seem to play that important role in binding calcium, so they don't seem to bind calcium in this case. Okay, right. Next up, we have a very important domain, which is going to form part of the catalytic domain. Okay, and I'm going to have to be careful because I'm going to run out of space if not. So the next domain is known as the X box. Okay, and this is going to be involved in forming something known as a Tim-like barrel domain, which we'll come on to in a moment, which is going to form the catalytic core that's going to catalyze the reaction, i.e. the breakdown of phosphatidylinosyl 4,5-bisphosphate into diacylglycerol and IP3. Okay, now, the other half of the catalytic domain is something called the Y box, and I think I'll put this over here to restrict how much space I can possibly spend on this intermediate portion here. Okay, to try and make sure that it all fits in. So here is the other half of the catalytic domain, and these together are going to form this Tim-like barrel domain, of which more in a moment. Okay, now, the portion then that's in between the X and the Y boxes, okay, is known as the XY linker. Now, in most phospholipase C enzymes, and I should stress this primary structure that we're looking at, these things that we're seeing, they are common to most of the phospholipase C enzymes. Now, what's different about the phospholipase C gamma enzymes compared to, for instance, the phospholipase C delta or the beta phospholipase C enzymes is they have an absolutely massive XY linker. Okay, and they have some really interesting domains and really important domains within this XY linker. Okay, so firstly, they have a split plextrin homology domain. Okay, so let me explain what I mean by this. So, these two portions that I'm now colouring in turquoise again, just like this plextrin homology domain that we saw over here, these are two halves of a plextrin homology domain. Okay, so together, these are known as a split, and then I'll just abbreviate plextrin homology to pH domain. Okay, right. So what do I mean by that? Well, I mean that when this thing folds up into its three-dimensional shape, this portion will become in contact with this portion here, and they together will make a domain that resembles this domain in plextrin, and therefore again is a plextrin homology domain. Okay, but they're not next to each other this time. The portions that are going to make this are split apart in the primary structure. Okay, so that's what we mean by split plextrin homology domain. And when I draw you the three-dimensional structure, or the supposed three-dimensional structure, the guessed three-dimensional structure, I'll show these two dimerize together to make a full plextrin homology domain. Now then, squashed in between the two halves of this split plextrin homology domain, you have three really even more famous domains than pH domains. Okay, so two of these, this one here and this one here, which I'll now colour in blue, two of these are what are known as SH2 domains. Okay, and this shows up all over the place in biology. Okay, so what does SH2 stand for? Well, SH2 stands for Sark homology domain 2, or rather Sark homology 2 domain, if you like. Okay, probably better to call it Sark homology domain 2. In fact, I will do that. Sark homology domain 2. Right, so the S is for Sark, the H is for homology, and then the 2 is for 2. Right, so these are, again, conserved structures that you find in a huge number of different proteins, and which were originally found within this protein Sark. Okay, so these are going to be extremely important in the activation of these phospholipase C gamma enzymes by receptor tyrosine kinases. So we'll come back to these. Okay, then, finally, you also have a domain called an SH3 domain. Okay, and this is another one of these Sark homology domains. So this one is Sark homology 3. Okay, now this one's not going to quite be as important as these two SH2 domains. And in fact, the two SH2 domains are so important that they're given separate names. So this one that's closer to the amino terminus than this one, okay? So you've got two of them in tandem like so. One of them is closer to the amino terminus, one is closer to the carboxylic acid terminus, which I haven't quite drawn yet. The one that's closest to the amino terminus is going to be called the N-SH2 domain, okay, because it's closest to the N terminus, okay, and the one that's closest to the carboxylic acid terminus here is going to be called the C-SH2 domain because it's closer to the C terminal. Okay, so that's just a little bit of nomenclature that will become useful later on, okay, because these are going to, as I say, be extremely important. Okay, so we'll come back to these two. We will draw the SH3 domain, but we're not going to see a great involvement of that. Okay, then you have one final domain after the Y box, which is known as a C2 domain. Okay, and again, C2 domains are very important in calcium signaling. They are domains that, for instance, you find in proteins such as synaptotagmin, and they sense calcium level. And again, they're not going to play a huge role in the activation of these phospholipase C gamma enzymes by receptor tyrosine kinase enzymes. However, there are other ways of activating phospholipase C gamma enzymes in which C2 plays a more important role. Okay, but we're not going to discuss those. So here is then the carboxylic acid terminus of our phospholipase C gamma enzyme. So that's our primary structure then. And the really key portion that's different from all the other phospholipase C families is this XY linker here. And I don't know why I'm highlighting that bit. Where's the XY linker? Here it is. Okay, so this portion here with the split plextrin homology domain with two SH2 domains, the NSH2 and the CSH2 domain, and then finally an SH3 domain there. Okay, so let's now put this together into a three-dimensional structure. Now, before we can put it into a three-dimensional structure, I need to talk more about these X and Y boxes here, okay, because these are going to form something called a TIM-like barrel domain. Okay, so I need to explain to you what a TIM-like barrel domain is. And in order for me to explain what a TIM-like barrel domain is, I need to firstly start off with what a TIM barrel domain is. Okay, so our XY portions are only going to form a TIM-like barrel domain. But we firstly need to explore what a TIM barrel domain is. Okay, now TIM barrel domains also have another name. They're also called alpha-beta domains, okay, because they contain alternating alpha and beta strands. Okay, and I'll explain this in a moment. So, right, let's see what a TIM barrel domain then is now. So basically, let's start with why it's called a TIM barrel domain. So the reason it's called a TIM barrel domain is it's found in a enzyme known as triose phosphate isomerase. Okay, now triose phosphate isomerase is extremely important in glycolysis. It converts dihydroxyacetone phosphate, okay, into glyceraldehyde 3-phosphate. It's that final step in the investment stage of glycolysis, okay, before you go into the payoff portion. Triose phosphate isomerase then is abbreviated to TIM by taking the T from triose phosphate and then the IM from isomerase. Okay, so that doesn't really describe what this is though. So let me now describe what a TIM barrel domain is. So let's start with the unfolded structure. So I'll start by just showing you the polypeptide structure on the page and then we'll fold it up into the barrel. Okay, right. So what you have is around 8 usually alternating beta strands and then alpha helices. Okay, so this is the amino side of the polypeptide. Now, because I don't want to just put the amino terminus there, because, you know, there could be all sorts of things prior to the TIM barrel domain. So what I'm going to do is use this notation where you put the amino terminus here, and then you put these two lines like so, which are put parallel to one another, and this means that there could be all sorts of stuff inserted in between here and here. Okay, and now we're going to start the tin barrel domain. Okay, so this strand here, this is going to be a beta strand. Okay, so it's going to be a nice linear portion of the polypeptide that can interact with other linear portions of the polypeptide and form a beta pleated sheet. Okay, so here's our beta strand. Then you're going to go into an alpha helix. Okay, so you're going to have alternating beta strands and alpha helices. So here's an alpha helix. Now you're going to do this 8 times. So here again is a beta strand, and then you're going to have an alpha helix. Okay, and then again you're going to do it again, and I'm going to have to try and make them a little bit smaller in order to get them all to fit in. Here again is our alpha helix. Then we've got a beta strand, another alpha helix. Okay, so understand what these beta strands are. They're linear portions of the polypeptide that are all running beautifully parallel to one another. And these beta strands, which I'll colour in orange here, are going to be able to interact with each other beautifully in a way that I'll show you in a moment. Okay, so let's continue this on. We need 8 repeats of this, so let me try and speed this up a little bit. So here's our next alpha helix, our next beta strand, our next alpha helix, our next beta strand, our next alpha helix, and then our final beta strand and our final alpha helix, and it just fits in. Okay, and then again I'll use this nomenclature. I'll put a splitting there, and then it goes off to the carboxylic acid terminus here. So there's all sorts of things in between there. Now, to avoid having to draw this out again, what I'm going to do is color code them. Okay, so in orange here we have the beta strands, which are all beautifully parallel with one another. Okay, so the polypeptides are all oriented in the same direction. So this is the amino side of this beta strand here. This is the carboxylic acid side of this beta strand here. And it's the same for this one. This is the amino side. And this is the carboxylic acid side, amino side, carboxylic acid side. So all of these beta strands here are running in parallel with one another. Okay, then we also have the alpha helices here. So in purple, these are the 8 alpha helices. Okay, and this is why this structure is going to be called an alpha-beta barrel. I haven't explained why barrel yet, but at least you should be aware now of why it's called an alpha-beta. Okay, because we've got alternating alpha helices. Okay, so here's an alpha helix with these parallel beta strands. Okay, so I want to now show you how this thing is going to fold up to form something that resembles a barrel. Okay, so basically what's going to happen is the thing is going to fold round and form a barrel, basically. So it's going to form a structure that resembles a cylinder, like so. Okay, so it'll have a nice sort of cavity down the middle here. Okay, and then the protein will form the outer aspect of this. And in principle, you know, you could walk through this and get out the other side. So what I now want to draw is this thing viewed from above. In fact, actually, I can probably do it on this. Okay, so what you're going to have is you're going to have the beta strands, the 8 parallel beta strands. They're going to be on the inside of this barrel structure here. So here are these 8 beta strands working their way around the inside of the barrel. And then on the outside, you'll then have the alpha helices. 1, 2, 3, 4, 5, 6, 7, 8, like so. And of course they'll be working their way all the way down like so. So here are these alpha helices. Okay, and I'll draw that one as well. And then the way it will go is that you'll start with this beta strand here. So it's difficult to show this now, but effectively the way it's linking up is this is our first beta strand, although of course it should be at the bottom here. So let me try and do this. Okay, so this is the first beta strand here. Then you go into this alpha helix, then this beta strand, this alpha helix, 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 this beta... Oh, sorry, I've gone a bit out of sync. This alpha helix, this beta strand, and so on, like that. And then you finally come out at this end here. Okay, so that's how it works. It's easier if I draw it from above. So let's draw the barrel from above. Okay, like so. Then we'll have these beta strands all lining the inside. I'll try and show these a little bit bigger this time. Okay. And then the alpha helices will be surrounding the outside. Like so. 1, 2, 3, 4, 5, 6, 7, 8. And the way this works is you start at the amino side with your first beta strand here, then you go onto the alpha helix, the beta strand, the alpha helix, the beta strand, the alpha helix, and this is our alternating beta strand, then alpha helix, like so. Okay, and then it will find the end. So that's how this thing is going to fold up into this barrel shape, basically. Okay, and when you have a structure like this, which is found within triose phosphate isomerase, it's called a triose phosphate isomerase barrel domain, or a TIM barrel domain, or you can also call it an alpha-beta domain. Okay, or even better, an alpha-beta barrel domain, we should call it. Okay, right. So, all of our phospholipase C gamma enzymes are going to contain a structure that's almost like this. It isn't quite as long as this, okay? Generally, you don't quite have 8 repeats of beta strands and alpha helices, okay? But what's going to happen is you're going to have repeats of beta strands and alpha helices within this X box here, and then you're also going to have them within this Y box, and these are going to associate to form at least part of a TIM barrel, okay? So it forms something that's similar to a TIM barrel, and hence it's called a TIM barrel-like domain. Okay, right, so we'll come back to this in the next video.